So happy to be the last presenter, and thanks for everyone sticking through it. This is Biocaml. The authors are myself, Sebastian Mondet, Philippe, Christophe, and Francois. Sebastian and Christophe are also in the audience today. This library has been under development sort of for 4 or 5 years, but really for the first 3 or 4 years of that, it was just my personal individual library that I was using at work. And only in the last year have we kind of picked up and developed a proper team around it and really sort of started developing it more maturely. When I got the reviews for this talk, which thankfully were positive and they wanted me to present, they really emphasized that I should explain biology because that would be new to the audience. So let me spend the first few slides just talking about biology. Now, if you look at the research budget in the US, half is basically spent on what we could call biology or life sciences more generally, and the other half is spent on everything else. Okay, so actually it can't possibly explain to you the whole of biology. It's a very broad field. What I will do is explain to you a very small subset of it, genomics, in a fairly concise fashion, and it's one of the more interesting parts of biology from a computer science perspective. It's really the part of biology causing a great deal of computational needs. So, okay, so this is what genomics is about. DNA, it's simply a molecule, right? And it's said to be the code of life because the sequence of DNA that each of us has really dictates a vast majority of our traits, in addition to, of course, environmental effects. If you just unravel the DNA a little bit, we can take the sort of simpler view of it, right? So the structure is double helix, but if you just unravel it, it's just 2 strands. The 2 strands each have a variety of individual nucleic acids hanging off of it, and they're different molecular structure, and there's basically 4 different types of nucleic acids. Okay, so and we indicate those 4 different letters: T, C, A, and there should be a G in there too, right? So if we go one level further, so first of all, roughly we can use that as our mathematical model, right? We want to model what DNA is, and to a great extent, just knowing that sequence of letters is a great model. That tells us a lot about what's relevant. Obviously not everything. For certain problems, you need to know the angle of the bonds and various other things, and this is not a sufficient model. But for a large number of problems, treating this as a string is a valid model, and that's what we care about. So, but even as a string, the structure of DNA gets quite complicated, and the amount of problems we need to work with is quite interesting. So even if you just consider a sequence of letters going from left to right on one of the strands here, there's a variety of like subsets within that string that have different properties. So some parts of that are exons, which are the part that turn into proteins. Between exons, you have introns. The entire gene is this part, and between genes, there's sometimes other kinds of what we used to think was junk DNA, but it's more recently being realized to have other functions that are not from genes. That gets transcribed into a different kind of molecule, which we still represent as a string, but changes a little bit, and there's this variety of structures going on, right? So it's strings with a variety of substructure that is of relevance, okay? So our goal is in Biocaml is to support operations on this kind of data, but it's not limited to that. It's just that that's kind of the problems where the people who contribute are working most right now. We can think of the main feature as being divided up into 3 major categories. We need to support file formats. There's a lot of file formats that biologists work with. We need some interesting data structures to work with the different data efficiently. And access to public data repositories is a really important requirement. And we hope to also add more and more interesting algorithms, but that's more future work. It's not something we've really done too much of yet. So we have a lot of file formats as a first support. It's sort of a core basic requirement, right? First of all, you got to read your data in. So it just ends up being the thing you end up supporting most at the beginning. Let me just give you a feel for the different file formats we work with. They're actually quite simple from a structural sense. The Fasta file format is a very common one, where the format is simply you start with an angle bracket and give the name of a chromosome. So we have multiple DNA on multiple so-called chromosomes. So you give the name of a chromosome, and then you give the particular string representing the sequence of letters occurring on that chromosome. No big deal, simple file format. The only issue is that the string is sometimes 10 to 30 million letters long. Okay, that's the issue. Right? Another format is, well, for a certain chromosome, from some starting point to some starting point, we did some measurement. We maybe measured the expression level of a gene at that point, and we got some numerical value. Okay, so again, not a big deal, except this file sometimes is 400 million lines long. Okay, that's the issue. A GFF file is one of these files that gives us this more complex structure. It tells us that from a certain location here to here on chromosome 1, we have something that is a transcript, and there's a variety of other metadata about it. Okay? So all these parsing issues, as you can see, it's not an issue of like having an interesting or complex grammar. What's really interesting is that it's just big data. Right? So one of the core requirements for us in all of our file formats support is supporting sort of stream-based processing. We don't want to load into memory unless necessary. We also try not to parse too much, even on a per line basis. If I don't need all the information, let's not parse it. And I'll show you some results of how much that helps us. We want non-blocking I/O. We want to do some error handling because inevitably these files are never following the specifications. And in fact, rarely do they even have a specification. So we can't really be too strict about the exact types that we expect to parse out. We're quite rigorous about documentation. I think the API documentation for Biocaml is amongst the best of any OCaml libraries. And we want everything to be zippable and unzippable and work seamlessly. So just to give you an example of one of the simple file formats, a Fasta module. Our Fasta module has this type called item, which represents the kinds of items that we expect a sequence of to occur in a Fasta file. The header is that name of that chromosome. And the sequence is usually a string, but we make it polymorphic because sometimes people use Fasta files to give something else called quality scores, and those are actually integers, so we end up having to use an int list. But we don't always parse into an item. We always define a raw item, which is sort of a minimally parsed item. And in this case, we just take each line that occurs in a Fasta file and don't really try to parse it into the higher item type that we expect. So you can have partial sequences where you just get the string on one line, which actually continues on the next line, but we only give you one line at a time. And the reason for this is some of the operations we're going to do, sometimes they're very simple. So you just want to count the number of items in your file. There's really no point in doing the extra parsing. And we did some measurements, and interestingly, we get some big speed up sometimes, right? 35%. If you want to count the number of items in a file, some of the files that I have are so big that that takes like 10 minutes. And if I do that over 500 files, and I do that multiple times, 35% speed up adds up to many days of compute time. So, for error handling, we've been using polymorphic variants a lot. This is sort of a style that we felt was compromise between just using a string for your error types and being providing more strongly like normal variants as types, which become a little cumbersome to use. We found polymorphic variants to be nice because for each type of error, so within an error module we have the error that can occur when you're converting a string to a raw item. These are the two kinds of errors. Since a polymorphic variant, we can quickly define them on the fly, and we can define other other errors, and then when you compose the two steps of parsing, with polymorphic variants, you can just take the union and you get the overall set of errors. So we feel like polymorphic variants are giving us fairly precise yet easy to provide error information. And we're kind of happy with that, but we're hoping to hear your thoughts also on whether that's a good choice. One thing we're struggling with is we're functional programmers. We like everything to be sort of type safe and nice, and we would like to use a result type rather than throwing exceptions. But on the other hand, it's our hope that some of the biologists that we work with actually will start using the Biocaml library, in which case, yeah. I don't know why that's happening.
Maybe the cable.
Yeah. Good. So we're kind of mindful of our target audience. One of them is us. Okay, we're all good functional programmers, so we can handle using an error monad to work with these types. But we would like to provide simpler, just exception-full interfaces. So most of our modules have a sub-module called exception-full. And if you open that, you just kind of get the exception-full version. And our intention is that's just for dirty scripting, where you don't care about catching errors. Right? So we're trying to balance the need for those different programming styles. Non-blocking I/O is important because all these files take a long time to work with, to read in. So the first issue is load or async, and you want to support standard I/O also. So our solution to this was to define something called a buffer transform. A buffer transform is something that converts alphas to betas, but also you can store some alphas. You can feed the buffer with alphas before you actually pull them out. So this is a feed function. Given a buffer, you can. I think I'm missing an arrow. There's supposed to be another arrow right there. You can give it an alpha, and you can read from a buffer, and you might either find out that you're done or not quite ready yet because the feeding hasn't provided you with the value, or you actually get the value. And by providing that simple API and then making sure we write all our parsers and conversion tools in terms of that, we're able to then wrap loop or async functions around it and have all things. But I know some of you have thought about this a lot more, like I know. So if you have any ideas on whether this is a good idea, again, we love your advice. This does seem to matter. We did a little bit of performance measurement on setting different buffer sizes, which is possible with the loop library. And for a variety of buffer sizes, we compared to just using standard I/O versus loop. And the main thing to notice is with standard I/O, you can't really get any performance difference. And then where with loop, you actually can do some tuning and affect the performance you get. Yeah. Okay. So we like nice composable components, and this is just an example of how we can do something nice. If we haven't quite settled on an infix notation yet for our operators because everybody keeps thinking about what that should be right. But let's just assume we use this for our compose function. This is a very nice example of how we can just create a parser from a zipped version of our Fasta file. You can just unzip it. We're linked to the CAML zip library. We can convert strings to raw items, and then we can convert raw items to full items. And the nice thing about this is if your file is not zipped, you just don't use that line. If you don't want to do full parsing, you just don't use that line. So we have the flexibility of what we want to do quite easily. Okay, so the next sort of big topic is data structures. Again, there's sort of no end to the data structure you might need in biology. We're really covering just a small set that we currently happen to be working with. And it primarily regards integer intervals. So let me just give you a few examples of the kinds of operations we end up needing to do. If you have a set of genes that you know about, and those genes have some coordinates on a chromosome, you know that this gene goes from location 100 to 150, this one goes from 200 to 250, etc. And then you do some experiment and you find that something is here. A very common thing you want to do is know, well, does this new thing I found, does it overlap any of the genes I know exist in this organism? So this is a simple overlap query you want to do. Again, it's fairly straightforward. The issue always is efficiency. Usually, this new finding, there's 100,000 of them per experiment, right? So we need to do this query 100,000 times. Read counting is another thing. When you do sequencing, you actually only get short reads. We wish you could just start sequencing the beginning of a chromosome and linearly sequence the whole chromosome. Unfortunately, that's not experimentally viable. The way sequencing is actually done is you chop up your DNA, and then you actually get short reads, and then you align them back to the reference sequence. So now, in this image what I'm showing you is short strings which we aligned to a reference. So we know that this string goes here, and this one goes here, and this one goes here, etc. Right? And then a very common question you want to know is, well, okay, at each location, how many reads did I get there? Okay, so just simply adding up the number of reads at each location. Again, trivial conceptually, except we often have 400 million reads, right? So to do this efficiently is an issue I'll show you in a second. Just one more example first. We often want to compute statistics like we do for receiver operator curves. The scenario here is you have a known set of genes, and then you do some experiment, and you find a whole new set of expressed regions, which you believe to be genes, and you just want to know how your new experiment compares to your existing knowledge. So you often want to do something like, say, compute the number of base pairs in my new region that overlap with the known ones, right? So those are false positives and vice versa. And you can see those are just set operations on integer sets. Okay? But this integer set has the property that most integers are contiguous sequences of integers. So it would be quite naive to store all the integers. Literally, you should use so-called diet sets introduced by Merritt Kurzweil a long time ago. Right? So, okay, yeah, skip that. And so to support the variety of operations we need on integer intervals, two quite useful partial orders that arise are what I call the positional and the containment partial orders. Positional just means an interval comes either to the left or the right of another interval, right? And in this, I consider U to come to the left of V, and also when they overlap, you still consider U to come to the left of V. Okay. The other relation is containment, which is just basically like a subset relationship. And if you merge those, you can get a total order. So using that, you can implement diet sets fairly straightforwardly. Actually, we're not using a tree implementation right now. We're just storing positional intervals in a list, making sure that the invariant of the list is every two intervals are positionally related, and there's no overlap. So it's maximally diet. And the efficiency of that is fairly clear in terms of memory. Obviously, it's like a 50x or 100x improvement in memory. Read counting can be done in a nice way, but there's one condition to consider. Oftentimes, we store the reads in our input files in sorted order. If your input is sorted, there's a low memory solution that's possible. It's quite easy to just keep adding up the reads at each base pair, and then when you get to a position I, you can print that value out and no longer keep it in memory. If you are looking at a current interval with a position larger than I, those are supposed to be the same I, got capitalized. But if they're not sorted, then you have to do something more complicated. You have to keep everything sorted in memory. You need an interval tree where the nodes carry the count, and it gets a little more complicated because you have to merge and split nodes as you insert additional values. Okay, public data repositories are pretty much critical to every biologist. You really can't do any work without it. In fact, it's virtually a requirement, I think, for every journal that data is submitted to a public repository, otherwise your paper cannot be published. And there's a wide variety of public data repositories. Entree, I have to say that biologists really have a very organized system for keeping data from their entire discipline. Not just experimental data, but data about their papers. PubMed is far superior to anything computer science has. You absolutely have a definitive source of publications if you want to go there. And Entree is the portal for that in the US, at least. And there's a wide variety of additional ones. So I'll just give you a quick demo to show you how we use that. We also have a command line app. And if you which is our way of encouraging biologists to use our library at first. Just give them a command line app, right? We don't have to know OCaml yet. We're using the core command API, which gives nice help documents. So you can just type help, and then there's subcommands. So you can say, okay, I want to use the Entree subcommand, which has another subcommand called PubMed. And then you can see, okay, PubMed expects a search term. So let's go ahead and do a search now. Let's say I want to find papers by me. And then you get some answers. And you get a whole bunch of answers because you find out that Agarwal is actually a very common last name, and most of us people are not. So let's add one of my co-authors who I commonly have published some papers with and try to narrow that down. And then you get some, these are all papers that are actually by me, but you still don't have any of the papers I care about because PubMed doesn't journal computer science papers. It doesn't index computer science papers, so the two papers I care about are not there, right? So, okay. So that's a rough overview. Going beyond what Biocaml currently has, I just want to give you sort of the opportunities for what's interesting in working in this field. We work with very big data. We have access to a lot of computing power. Amongst that includes GPU nodes. I have no idea how to use them. So if anybody wants to work on a project to do very interesting parallel computations on GPUs, there's clear evidence that this would be highly useful to biological problems. There's another software called Bioinformatics, which has demonstrated this. The software engineering behind these tools just isn't good. Nobody in biology is really using these hardware because the software just aren't nice enough and don't quite work. It's hard to compile them. I think we can fix that problem and show some genuine value here. So in conclusion, basically all aspects of computer science are relevant to biology. I think the old view of scientific computing is it's all numerical computing, and that's just not true anymore. Numerical computing is highly relevant, but I basically don't need that at all. What I need is string manipulation, interval trees, XML parsing. I need to make a website for my collaborator. Those are the things that that scientists really need these days. It's an exciting field to work in the US, which is not a model of healthcare, but as an example, in the US, literally 1 out of every $5 is spent on healthcare. And it's a bit depressing. So if we can make the field even like 1/100th more efficient, that would be a dramatic contribution to society. So you can find more information at biocaml.org. We have a mailing list. We have everything on that side. Okay.